So what if I told you there's a new way to organize the very building blocks of life? I'm not talking about just the long list of parts, but a structured, predictive map. Kind of like that periodic table of elements you probably memorized back in high school. But to really get how we arrived at this new periodic table of cells, we have to start with a question that sounds simple, but is actually surprisingly tricky. We're going to kick things off with a question that sounds so basic, you'd think we solved it centuries ago. What is a cell type? Well, it turns out the answer is way more complex and, honestly, way more fascinating than you might think. You see, for as long as we've been able to see cells under a microscope, we've been trying to name them and put them into little boxes. But this has created a huge problem of identity. We've ended up with this messy, unstandardized system that's frankly more like a chaotic stamp collection than a real scientific framework. Just look at how our definitions have changed over the years. It all started way back in 1665 when Robert Hooke first looked at cork and just saw cells. Then, for a long time, we classified them by shape. Is it round? Is it star-shaped? Then we got a bit more sophisticated and started using function, like T cells, named because they mature in the thymus. And more recently, we've gotten really specific, defining them by a handful of marker genes. Every step added more detail, sure, but it never gave us a single universal set of rules. And that really gets to the heart of the issue. We've wound up with this gigantic catalog of cell types, but we're missing the instruction manual. We just don't have a formal way to understand how they all relate to each other, how one can turn into another, or even how to organize them in a way that makes logical sense. And then, a technology came along that completely changed the game: single-cell RNA sequencing, or scRNA-seq for short. And this promised to finally bring some order to the chaos. For the very first time, instead of just looking at a blurry average of thousands of cells all mushed together, scientists could pick out one single cell and read its entire genetic activity, its unique fingerprint. It gave us this incredible, high-definition picture. But it also uncovered a brand new and even more confusing problem. In some tissues, like, say, the adult pancreas, the data was just beautiful. The cells formed these neat, separate little islands on a graph. You could literally point and say, okay, that's a cluster of beta cells, and right over there, that's a cluster of alpha cells. It was simple, clear, and easy to classify. But when scientists looked at systems that were developing or changing, like sperm cells maturing in the testis, they saw something completely different. Instead of those distinct clusters, they saw this continuous, flowing river of cells. There were no clear boundaries at all, making it pretty much impossible to draw a line and say where one type ends and the next one begins. And this led to a really deep conceptual problem. Is a cell that's halfway down that river its own unique type? Or is it just in a temporary state while it's on its journey to becoming something else? Suddenly, our old definitions just completely fell apart. So how in the world do you solve a puzzle like that? Well, the researchers behind this idea came up with a new framework. It's all about separating a cell's deep, fundamental identity from its temporary behavior. And you do that by focusing only on the genes that really, truly matter. They suggested we should focus on something called the core regulatory complex, or QRC. The big idea here is to ignore all the noise from thousands of different genes and instead look only at the master switches, these things called transcription factors. You can think of them as the bosses of the cell. They're the proteins that control which other genes get turned on or off. And that's what really defines what a cell is at its core. So here's how their approach works in practice. First, you analyze the cells the old way, looking at all their genes. And that gives you a messy map that has both the stable types and the temporary states all jumbled together. But then, and this is the genius part, you do it all over again, but this time you only look at those master switch transcription factors. And by comparing those two maps, you can finally separate the signal from the noise. And this whole process gives us two really clear, really powerful definitions. A cell type is its core identity, defined by that stable set of master control genes. It's who the cell is. A cell state, on the other hand, is just what the cell is doing at any given moment. Is it dividing? Is it stressed out? Is it responding to a signal? That's just a temporary program running on top of its core identity. So once they had this powerful new way to define cells, the researchers had their big aha moment. They realized they could finally organize all of biology's incredible complexity using a simple, elegant, and really familiar analogy. The big solution was to create a periodic table of cell types. The idea is Just like the chemical table organizes all the elements based on their fundamental properties, this new table could organize all the cell types based on their core identities and how they're related through development.
Okay, let's break this analogy down because it's just so elegant. The elements, of course, are our new clearly defined cell types. The rows of the table, or periods, represent entire developmental journeys, like the whole pathway from a single blood stem cell all the way to a mature red blood cell. The columns, or groups, line up cells by how mature they are. So you'd have all the stem cells in one column, all the fully mature cells in another. And what about those temporary cell states? Well, they're just like isotopes, slightly different versions of the same core element.
So this is obviously more than just a clever way to make a chart. It's really a blueprint for biology. So now that we have this framework, what does it actually let us do?
Well, for the first time, we can logically organize every single cell in a species. We can finally see the exact relationships between different families of cells. We can even compare these tables across different species to understand how life evolves. But the most powerful thing, the real game changer, is its predictive power.
Think about it. When Mendeleev first drew up his periodic table, it was full of gaps where he predicted new elements would one day be discovered. This biological table has the exact same thing, empty spots. And these gaps aren't mistakes, they're predictions. They point directly to where missing link cells have to exist. And the paper gives a perfect real-world example of this. In the brain, we know progenitor cells develop into neurons and other things, but how we get ependymal cells, which makes spinal fluid, has always been a bit of a mystery. Well, the table's structure basically screams that there should be an ependymal progenitor cell filling a gap in the chart. It gives scientists a specific, testable hypothesis and tells them exactly what to go looking for.
What this all means, at the end of the day, is that biology is fundamentally changing. It's moving away from being a descriptive science, just cataloging a list of parts. And it's becoming a predictive science, with a logical framework that can actually guide us to new discoveries.
And that leaves us with one last, massive question. If we can truly map every fundamental element of life and understand the rules that connect them all, what new medicines, what new therapies, what incredible discoveries are we going to build? The possibilities really are just staggering to think about.